Okay, so let me share my screen. Okay. Think we have forum. Yeah. Okay. So let's get started. Well, again, we're going to record this meeting and share it broadly. If you have concern with us doing so, please send us an email after the meeting, and we will refrain from doing that. But just wanted to acknowledge it. I have the meeting notes here as last time. So last week we met and discussed the overall framework, how we want to track this project, and we said we would go through a weekly cadence where we'll alternate between stimulus design and analysis. And so this week, I think we want to cover the stimulus development. And I was hoping we discuss a few things. So first, I like to introduce a little bit resources on bonsai development that I've started to put together, and I think that I've had it since last time. And then I like to sort of highlight the progress on the old ball stimulus for the standard old ball, and then we'll see if I can show the bonsai workflow, and then do the same for a sensory motor stim, so that we can discuss both of these. And then if time permits, we could discuss the duration old ball and have a discussion about how to replicate boost workflows outside of the island so that everybody can weigh in on the stimulus parameter. Uh, does that sounds like a good plan? Any things we should discuss before that? Okay. Um, so I think I introduced a little bit the bonsai resources last time, but I want to facilitate this because bonsai is a programming language, and many might not be familiar with how it works. At the very least, I think it would be beneficial if a number of lab run our bonsai workflow independent of the hardware in the lab. So I think that's totally possible where you pull a bonsai workflow and then run it on your computer to see how the stimulus unfolds. And it would be beneficial because I think a couple of Visual Lab could provide feedback on the parameters, and we make sure that what we are planning to run is well grounded. So in the stimulus section here, we provided a number of things. So the first thing is an introduction to bonsai. Uh, so this is if you want to do programming, and I collaborated with a couple of people, and notably Bruno Cruz at the Allen Institute, to sort of highlight what are the good resources to learn how bonsai function. And so I recommend this introductory bonsai video by Gonzalo, the main developers of bonsai in England. And then if you do that, and that's a fairly long video. And then there are also other learning resources in the documentations of bonsai that I think are worthy looking into it. The first thing is if you go here, this is the documentation of bonsai, and there are a number of things that are really worthy. So the first thing is this language guide here that highlight how components of bonsai work together. And so it 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 introduced the reactive programming aspects of bonsai, which is I think unfamiliar to many. And so I provided links to these resources, which I think are probably the best documentation there are from the moment. In addition, we also added a little tutorial for if you're a Python programmer, which I think many of us are. It's a helpful bridge that Josh wrote that introduced the different objects in Bonsai and how they work with one another. Especially, he talks about for loop, if control statements that are absent from the language, which is kind of confusing initially. So you have to think a little differently when dealing with Bonsai. And so I recommend this is a good read. It's actually quite short. The 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 Bonsai for Python programmers. So there's the YouTube video. I think the documentation. So this is a longer commitment, but I think it's useful. And then the Python programmer is shorter. And then for replicating our workflow, we wrote something a Bonsai instructions, a markdown that explain how to set it up. And the core of it is that in our repository that we share, there's a little command line called setup.cmd. And on our Windows machine, you launch this command. And what it does is it downloads all of the executable that you need to run the script. So it recreates a local bonsai.exe along with all of the packages that this stimulus needs to run. And then you go to the local bonsai folder that it created and then launch the bonsai executable. It's explained here. And so that essentially guarantees that you're using the same package that we are using on the experimental rig, and it should limit the problems with replicating package versions. And so I think what will be fantastic is if a couple of people go through this and try to run our first version of scripts to visualize the stimuli on their screen. So that's why we did this. Have any questions on this? Yes, Alex.
Yeah, so as a vision scientist, the one concern I have is that each screen, every monitor is different. So maybe we should add something on gamma correction, linearization
Okay,
to make sure.
that's a great point. So now that you're mentioning this, so in the project tracking, we have a project board here. I need to update the link. If you switch to the board view, you'll see the things we are working on currently. And my first top of the list item here is to deploy a gamma calibration bonsai workflow. And the way it's going to work is we have a little photodiode at the bottom right corner of the screen that is linear, and so it measures the light coming from the screen. And Bonsai has a couple of, I think you know, Alex, Bonsai has a number of workflows that do gamma calibration. And what I need to do is commit this workflow to the repository after I've confirmed that they work as advertised. And so that's actually my focus this week as part of the SLAP experiment.
So secondary with that, I think maybe we should also be very specific about the exact eye to screen distance to make sure that, or build something in that if you have a different eye to screen distance, that the stimulus scales the same way to have spatial frequency and stimulus size also norm.
That's right. So let me actually, I think I need to show it because I think it might be worthy that we discuss it with the Bonsai workflow on the screen. So I'm just gonna remote connect on a computer on a Windows machine. Because the only thing with Bonsai, it only works on Windows. Okay. Let me just load. Just loading the bonsai script. I'm just gonna pull it. Sorry, just bear with me for a minute or two, because I think it's gonna be useful. Okay. So if I stop sharing, it should work. Share. Okay. Can you see my Windows machine? Okay. So this is the repository that is cloned on this Windows machine. So you could pull it. And then this is a stimulus control folder under the repo. And then you will see in sources the script that we are using. And in Bonsai, there's this little setup command here. And so what I'm asking everyone is to launch this little setup command. And what it's going to do is going to actually download this executable locally, along with all of the package folders. So this, if you pull the repo, these are not present. You will only see, I think, NuGet config, setup command, and setup.ps1. All of these, except maybe I think the shaders are not on the repo. And so it downloads them. And then once you have downloaded them, you just launch Bonsai executable. You double click on it. And then you go into the folder, and hopefully I just pulled the repo, so hopefully it's not going to break anything. But and you open this up. So you see I'm opening the standard oddball on Slab2. Okay, so this is the things I wanted to work through a little bit with you. So this is the first version of the script. And at the top one, actually the top object here is called create window. And so you define parameters of the screen here. And I think where is it? It's in Bond Vision. There's a position where we create the screen geometry. I'm missing it. Oh, I see. So this line here, render frame, orthographic view, and view window. So view window here, define. There's a whole documentation on the Bond Vision website that we can link in the web in our website. So here it sets a translation. So it positions the viewer 15 centimeter from the screen center. And you also set the dimensions of the screen in that 3D space. So whatever geometry you're using on your own experiments, you just need to input it in that object very carefully so that the spatial frequency is correct, as Alex mentioned.
So, Jerome, my point was that maybe as we have this quasi tutorial now, that we maybe even in red or with an exclamation mark, put that in there so that because somebody might forget or not know about that, that we can exclude that after we collect data, we find out we didn't collect data to the exact same stimuli, and that could become a
Yes,
problem
yes,
peer
yes.
review.
There are many mistakes that could be made on the way, so we should be careful. This is partly why I want a few labs to download this and run them on their labs so that we double check each other's work so that there's no mistake. Yeah. And so I feel like if you could do that, Alex, that would be wonderful. I don't know if other people in the call will be interested in helping out with this validation. Um, If you're interested in that, please raise your hands. I would gladly help you out. I know Sarah has already started. Okay. Um, so the next so I let me explain a little bit how this workflow works. So because I think that would be helpful. So this script, well, actually, so if I want to run it on this Windows machine, I need to disable the behavior device because they are not available. So I disable this device here. And I also disable the behavior cameras that are at the bottom because this bonsai workflow record behavior videos at the same time. And I think this should run. Okay, so it's currently blue because Slap2 is currently only compatible with blue stimuli. And so you see it's currently waiting for spacebar to start the experiment. And there's this little photodiode at the bottom right corner that flicker. Just because on the experimental computer, there's a photodiode in front of this that we track precisely. So if I press space, it's going to start the gradings. This stimulus start with orientation tuning blocks. So this is randomized, so you see drifting gradings. And this in this particular configuration, this is a quick demo. So now we are already into the oddball block. So it's you remember the design. The drifting gradings is only in one orientations, and then it repeats and then introduce oddballs midway. We already finished the oddball blocks, and now we are back into orientation tuning.
Okay,
And
yeah,
then
Jerome, can I
yes,
stop with here?
yes,
So
please.
one thing that I spot, again, as a vision scientist, you would probably not use a blue on black contrast. And that's because you're changing both contrast and brightness of the whole image. So
Mm-hmm.
what is more typically done is that you have a neutral gray, maybe even something like 50%, and then the gradings are on top of that. So then the local minimum and maximum of the grading cancels to the neutral gray.
I think
This could
that's the
this could be
case.
the previous one seemed. Oh, okay, I see.
I think
Okay,
that's the
never mind then.
case.
Never mind.
Yeah,
Yeah, okay, I see now. So that blue is the average value of the
I
blue
think
and the
yes.
high blue.
Well,
Okay, good.
provided we do gamma calibration correctly. Um, okay, so let me replay this. So you see, we start with orientation blocks. It's fairly quick. In the real experiment, there's a repeat number that is increased a lot. So and then now we're already oddball blocks. So it's right to left. Then this is an oddball. This was an omission actually. Um, and then this is another oddball. It's a new orientations. Um, it's this gonna. I think there's another. Yeah, this was just two oddballs in series. Vertical and a halt. And then yeah, now we into the orientation block at the end of the experiment, and then we do receptive field. So this is a very simple first pass at the standard oddball. Okay, and we have run this script on Slap2 3 times, and last week we confirmed that we do see receptive field, beautiful receptive field of boutons in the dendrites with the Slap2 experiments. Okay, so you can abort anytime. So there's a couple of things that I want to highlight here. So these here is Bond Vision cooking. So it's creating the window. The shader is for creating the blue screen. And then it's sort of assign resources to the system. This lines here is drawing the photodiode. And a bonsai is reacting programming language. So it you connect objects that are meant to respond to one another. So the draw here is something that every time a draw is done on a screen, it sends an event. And so This is connected to the DrawCAD to tell that we want to draw a photodiode corner in the bottom right. So you can have many objects connected to draw in different lines. So it's just the way bonsai work. This line
can I
here,
ask,
yes.
Jerome, just a really quick question. This is a little bit of a detour, but I already looked at the slab data. Did you also have a photodiode to synchronize the neural data to the stimulus events in that case?
We have we're using a digital output port. We're using a digital port to that is I can show you later on. That is being switched whenever the grading starts. But it's not, yes. So essentially, yes. So it's a bit of a hack, but it's working. The thing with the SLAP2 data that you looked at last week, Carter figured out there was a bug in the temporal alignment code. And so we have a new NWB to push that fixed that alignment issue. And you might see a lot more tuning with the new files. Okay. Any other questions?
It's just
Yes.
really quick because I did run into that issue. So the event trigger in the end, that is just a regular part of the NWB. You don't have to look for something special in the NWB file to
No. The
see.
time in the NWB, we align. We in principle everything is aligned to the same time clock. And so that's why if the alignment is wrong, then you're stuck. So we have to fix it. It was actually a funny MATLAB to Python bug. So the alignment was done in MATLAB. The first version of the alignment was done in MATLAB, which is 1 index, and then Python is 0 index. And so we just were assuming everything was 0 index by mistake. Okay, so the next line here is SLAP2 related. So this is just sending pulses to SLAP. So in the context of stimulus design, you can ignore it. Same as this one. This is sending pulses to start the microscope. This is doing the wheel reading, so reading the wheel encoder for the mouse. And so you're going to see that it's being used for the sensory motor oddball task. This is saving the encoded data with timestamps. So it's timestamping the values so that we can visualize it. This is the core of the HARP ecosystem. So it's taking commands to the card and then sending responses to behavior events. So the port name here is the COM port of the HARP board that we have on the rig.
Can I jump in here real quick again?
Yeah.
So I tried to look for it and I couldn't really find the exact hardware specification because this suggests to me that we all should buy the same hardboard and use the same hardboard.
Right. So we actually had a couple meetings internally this week. We are in the process of changing the hardboards on SLAP, and I'm hoping that we'll write a page on this in the website because I think there are several cheaper boards that we could point you at. For instance, we have a new TTL board that is $100 to purchase that could be helpful for your case. Okay, and so I'll write down this hardboard, but in the interest of replicating the stimulus, you can implement your own hardware in Bonsai. And so I'm going to show you one thing that I'm doing to deploy this stim on the rig that is not using HARP. Yes, thanks. It's a really good question, Alex. Okay, so this is again related to SLAP. And so it's just doing stop at the very end of the experiment. When you connect something to workflow output here, it closes the stimulus script. So this is telling it whenever someone pressed a key, which is in this particular case end, and the experiment is finished, you tell SLAP to stop acquiring data, and then you tell Bonsai to stop. So this is the end. So you will see that bonsai is just it's a lot of threads that are in parallel, and so when you have to understand row by row. Um, this is about logging. So it's just how we log the behavior events. This is to generate the folder where we save the data. So we use some logic that is using the mouse ID to create a local folder whenever the experiment is started. That row is as we said, setting up the geometry. So that's very, very important here to do this right when you're setting up on your machine. So you have to understand how the view is created, how this is mapped into a sphere, and then how you create the view. Yes, Ahmed.
Uh,
Yeah.
Yep, I had a question about the columns. Is there any specific like relation between columns, or it's
No.
just it's just row by row?
So Bonsai is visual programming language, but whenever you had a new object, it reorganized the view a little bit. It's just you create connections and so you say you click this object, say, and you right click, you say create connections, and then you select another object. Say let's see something that could be useful. So if I say create connect, if I insert a new object, let's say I create a draw. So you see, when I click a draw, it goes at the bottom. And then if I say, um, I'm sort of repeat. So you see, it had it, had it. So you can either write it down here, or you can drag it from the list. So if you do that, it's also work. So it essentially it's you sort of drag object into the GUI,
And
and then
another question I had
yes,
was about the stimuli. So in Bonsai, it renders at the time and presents for
yeah.
certain stimuli. In Monkey Logic, we load and play like the stimuli is pre-made.
Oh.
And one of the questions we had was like, how necessary is it to switch from Monkey Logic or like change from Monkey Logic to Bonsai for people who use Monkey Logic?
Well, so what I'm trying to do, so all the experiments at the Allen will be done with Bonsai, and I I think all of the hardware to do recording, all of the hardware to do temporal alignment can be different, but I'm a little anxious that the stimulus that we run is the same if we want to integrate dataset across. And so I think using the same software might make sense, which is why I'm doing this to sort of help out how bonsai work. The reason we chose bonsai at the island is that it's actually quite a powerful programming language, and it's very, very good at doing this real-time adjustment. Um, and especially for closed loop task. And since we have a sensory motor task that involve closed loop design, this is superior language to do that. You'll see that in the sensory motor oddball, doing this loop closed loop with the wheel is like super easy. And it's also very good. It's all C sharp under the hood, so it's actually quite efficient. And so I think it has a number of advantages in that realm. Um, and I'm sure there are other solutions, but if there are one place where we might benefit from integrations is I think in the stimulus code. So that's why I'm advocating for it. If you really don't want to do that, I think we'll have to be really careful that the same thing is run and displayed. And so it's another piece of work.
Because I mean, changing a stimuli and aligning them is easy, but because our setup is like both rigs are on Monkey Logic, and we have like two lasers each side, so we
I
have to
see.
re-implement all of our pre-processing and stuff like that,
But
and it
which
would take
which
about.
pre which pre-processing?
So like instead of Monkey Logic, it has to be Bonsai that sends the digital codes, analog codes, and all of the laser control, the simulator, and stuff like that. So
this is for opto tagging, or
yeah.
um yeah, I mean I guess, are you doing this during the experiments or at the end?
During the both
But it's
kind of like
intermixed.
during we have a task that is during the experiment, but usually we do it in the morning to tag the nodes.
I see. Yeah, I think there are different ways to do it. I'm pretty sure you can do this in Bonsai. Um, we can discuss. Can you create a thread on the forum to discuss that?
On GitHub, yeah.
Yeah. Okay. Um, then so this is the lines that set the screen, and then the core of the experiment is this row. And so this is concatenating 4 blocks. So you see there's a concat object, and it's actually quite simple. So your grading specification, if you double click on this, I have a list of orientations. So this is a list. It's a list of objects, and then this is the orientation is selected, and it's creating a bunch of grading trials. So there are default parameters at the right here. So we are just changing the orientations against the default sets of grading values. And then this is repeated a number of times. So this is the orientation tuning block. And so the repeat count is provided by this parameter here, which is workflow input. So you see the number of mismatch per conditions is actually a value, is an input value of the whole workflow. So whenever you start the experiment, you can change this variable to like 100, and then the experiment will be 100 times longer for the orientation tuning. And then there's a permutation. So this is essentially creating a list of sweeps and then permuting them so that they're randomized. So that's the first orientation block. Then this one is the baseline. So there are where, how does it work? Yeah. So this is the baseline grading. So it present the baseline of the old ball, and here these are the odd balls. So there are 4 of them. And so they are both permuted and then concatenated. But you see here that there's a merging step, which means that the grading specifications and the baseline specifications are essentially merged eventually to create the mixed sequence of oddballs and baseline. So that line is the most important line. This is where the oddball blocks is created. Then this block is the second orientation block at the end, and this one is the orientation tuning. It is the receptor field mapping, and this one is a little different because it's taking a CSV files as input to contain the list of locations because it's very long, and I found it was simpler to use a CSV. So essentially, there are 4 blocks. First, orientation. Second, oddball block. Orientation again, and then receptor field mapping in this particular iterations. And then this is passed down in order to the display for display. So all of this sequence here is to display one by one all the drifting gradings. And so at the end, when everything is consumed, it creates this end experiment object that trigger the end of the workflow. And then the bottom here is all about logging the information in files so that we can keep track of what happened. So that's the core of this design. And I think it's currently functional, and so we've been using it for the SLAP2 data. I've questions on this part.
So at the end, it spits out a CSV file.
All right. So at the end, the Bonsai script for us save I think 2 CSV files. One with the list of gradings and all the parameters that were displayed. And a second one, I think, with the frame number that across times. It also saved a bonsai file, a harp file. So this is using the harp card that is a list of a lot of binary single files that store the encoder and all of the pulses that were recorded by harp. So this is like two systems for tracking. One is using Bond Vision and the other is using Harp, and they actually duplicate a little bit the tracking so that we can debug if there are any issues.
And then I assume you have some kind of code that takes that and combines it with the neural data to create an NWB file. Is that correct?
That's right. So that code is what Carter is working with on CodeOcean. These are capsules that we will share that takes the input file and then create the NWB after after some processing. So for SLAP2, Casper's team has a bunch of MATLAB script that creates an intermediary data assets that we read in Python and then create the NWB. And ideally, we share the processing code, but you don't need to interface with it. Because it's the same thing for NeuroPixel and Mesoscope. They each have their own processing pipelines, and I would like to share NWB files with you guys so that you don't have to know about each rig details. Okay. So if what I was planning, so currently the standard oddball has the rotation block, the oddball block, and then the receptive field mapping block. This is not it's not 100% exactly the same as our final design that we had in the document. I think if you look carefully, it's not exactly. This is tailored for SLAP 2 at the moment. And once we migrate this to the NeuroPixel rig, I'm planning to sort of follow exactly what's in the document. But it should be a simple thing because it's just blocks that you move around and parameters. So I'm not too worried about it. But that's where we are in the standard oddball. So I think it's fairly close to being fully implemented. And it would be beneficial as some of the lab here could pull the Bonsai workflow and run it so that we could confirm that this is good. Any questions? Okay. The second thing I wanted to show was the sensory motor all ball stimulus design. And I think you can still see my window screen. So it's another file that is here. Okay, so you're gonna recognize a little bit. I'm starting to structure it a little better. No, sorry, it's the wrong. Oh, that's the right one. Okay. Let me just remind myself how it works. Right. So I started to structure this a little better and added group of bonsai objects so that help reads. So for instance, the photodiode rows are now contained within their own subgroups, so you don't have to look at it. The sensory motor coupling is implemented here, and so if you just double click, you go into a sub part of the workflow. So you're going to recognize some pieces. Where is draw. So the draw grading is here, and that's the beauty of Bonsai is implementing the coupling is actually as simple as having the temporal phase coming from another object. And so this here is just doing the coupling, and the temporal phase is coming from the encoder. So the encoder provide a speed value, and then this value is divided and go through a modulo operator so that we are converting this to a phase. And then this phase is fed to the draw gratings. And so suddenly now the wheel controls the gratings real time. The way oddballs are implemented is currently is actually I keep this system running, and then I display another gratings on top of the gratings on the screen. And so this object here is essentially randomly at any moment in the experiment display a second object, visual objects on top of the grating that is coupled. I tested this during the experiment during a session, and it functions. So essentially it pulls a random time, and depending initially at the beginning of the experiment it creates a list of random times at which this should occur, and then display either gray screens or gratings of different orientations on top of the existing grading. So that's how the sensory motor oddball is implemented. So quite simple.
Jerome, can I ask you quick? Would it be possible for where you take the output of the ball and you feed that for the spatial phase? Would it be possible to multiply that?
That's here. So there's a divide value that we could change. I mean, it's a division instead of a multiplication. I haven't figured out exactly the gain I want to apply. I think, yeah, I think you probably want to be smart about this so that I mean, we'll probably follow what Keller has been doing in terms of the baseline speed, because you probably want to have the speed of the gratings, the natural flow that the mouse experience when it's walking forward, as like I would say the most natural gain, and then play with that from that as a baseline. That's my intuition. But it works. Yeah.
Jerome.
Just a quick, alternatively, it may also be interesting to just show a static grading, a non-moving grading that would, it's not as perfect, but it would also achieve that effect. That there's two parameters here. One is the fact that the gratings are moving, and then we discovered that from the running speed. But it would also be interesting if the running speed, if the translation, if the gain is different, that's a different oddball, and that raises interesting questions. And so the extreme case would we stop the grading, right? So in that case, that would be that the gain is completely out of whack. But it would be perfect if we can parametrically vary that.
And that would require a slightly different formulation than this, right? Which just takes the encoder position and divides it by a number and does a modulus. You'd probably have to you'd have to accumulate and keep track of a phase, but that would be very easy to program. I have a sort of, sorry to interrupt, I have a similar sort of thought, which is, your oddballs are random phases, Jerome, but I'm not sure how salient different static phases are in this motion thing, where it's really the visual flow that matters. So picking, setting the phase 0 and putting a flat phase 0 probably doesn't look that different than putting a flat phase 180 on the screen to the mouse. It's just suddenly the movement is decoupled. Would be, I mean, I don't know, but I'm just saying, is that really the salient thing, or would, for example, changing the gain, right, be the more salient oddball manipulation here?
Yeah, I think we were initially planning for 4 types of oddballs: an omission, so gray, an alt, so the grading suddenly becomes steady, like it doesn't move anymore, even if the mouse is moving. And two alternative orientation tuning. So like instead of horizontal, it would be like 45 degrees and 90 degrees drifting gratings, which is like very sudden. Um, the idea was to have the exact same oddball in the standard oddball block, so that you'll be analyzing the same experience but in different context. Uh, I don't think we have considered adding a different speed. Uh, we can definitely do that. We just have to work out how many trials we have if we had a fifth oddball. Um,
So
I
to
mean,
clarify, Jerome, so the oddballs that you're showing are actually just drawn from the same oddball distribution as the other experiments.
they are the same.
So they are drifting gratings of different orientations or blanks.
Yes.
Okay, sorry, I misinterpreted the bonsai workflow. Thanks. That kind of makes sense.
That's the idea.
So the one thing that I'm interested in is if you have two oddballs. One is that this coupling is complete, so there's still optic flow, but the mouse lost all control over it. Versus the mouse has still some control, but it's still an oddball because the speed is off. It's too fast or too slow. Then we're having a very, very, very interesting new experimental paradigm where prediction is not just a pairwise conditional probability. And so it opens up a lot for predictive coding if we can get this implemented. It may be well worth doing.
Yeah.
So it's getting into higher order predictive coding.
I guess what we could do is instead of having two alternative orientations, we have one alternative orientation and one speed.
Yeah, half as fast or something like that. That would be very interesting. Yeah.
Yeah, but I mean that would mean in the standard oddballs, I don't know that the half as fast is. Maybe we could do that, but can you create a thread in the forum so that we could discuss because I feel like there are ramifications on statistical power that I want to think through. Like it seems like you're making a case for that. I think Kareem also was making the case for that last week. So I feel like there's enough value in doing it. So I'm fine. Yeah.
Yeah, this pertains to a discussion you and I had in previous meetings, where the primary thing, as Alex said earlier, that to control the gain, have control over that gain, but then we could also explore the changes in that gain within the same sensory motor paradigm, right? So it could be smooth, right? So the animal gradually would see or would witness or perceive a change in the gain gradually, or it could be abruptly, all of these scenarios are different variants of the same thing, but that's a most interesting question to me, at least.
Yeah. Is that something that you could do in your lab? Like we could do the change of gain as part of our experiment, and because I'm thinking that like slightly smoothing, changing the gains or things like that will be a lot more. It might be more experiments. Like you might want to focus on that problems and have.
Yes, it is because I'm interested in how dopamine influences movement vigor. That's one specific question that I'm interested in. So that's why the sensory motor makes perfect sense, because dopaminergic neurons have typically shown this ramp, right? And there's a lot of debate in the community regarding, what these ramps mean. Do they mean approaching reward? Do they mean, speed? Do they mean other things? So this is a paradigm where one can investigate this to see the difference between the gradual ramping of the gain versus an abrupt change in the gain, right?
Yeah.
So,
okay.
Yeah, we can certainly explore that. Yeah.
So if you okay, so that brings me to one thing that I wanted to discuss related to replicating this sensory motor. So let me go back to the website. Okay. Okay, so in the project tracking, if you go here, So I really need to change that link. It's not convenient. Okay. So this is what we're working on. So we are laying out some internal document to track the experiments. That's will be shared eventually. So I'm working on the first version of the mismatch and the first version of the sensory motor. And last week I started to pilot this on the eFish rig, which made me to actually investigate how to do the wheel encoder in Bonsai. And so I'm currently working on Bonsai modules. And so it's a repository that you can find here that is essentially reading the encoder data for on the different rigs. And so I think this experience is going to be helpful for others. Because in the Bonsai workflow, you can replace the encoder object with whatever hardware you have to measure movement of the mouse. And so I'm implementing two. So we have one with harp, and now I'm doing one with an Arduino card that is used in other rigs. And so for hopefully that experience will be helpful for other labs to modify those workflows. And I think in your case, Karim, that might be relevant.
Yes, thank you.
And then the next, and these are the current tickets we're working on. So Carter is going to update the slap experiment with the better timing. So Alex, you can track these here. Because in next week we want to discuss the slap analysis in more depth. We have some really cool receptor field plots that we want to show. And I think that can trigger an interesting discussion for next week. Okay. Which one I wanted? Okay, so then I don't know if Sarah is on the. I think she's usually in the chat. Okay. Oh, she wrote a couple comments. Watch. I did. Okay. I don't know that Sarah is going to be able to sort of. So Sarah started to work on the direction oddball, and I think we need to test it at the Allen. So I will do that in more depth in the next 2 weeks. But she's in the so she's in the tube. Sorry about that. Okay, so can we spend a couple of minutes discussing how we can replicate those bonsai workflows outside of the Allen? I think Alex, you already started that, or
Yes.
okay? Can you focus your attention on the standard oddballs and give us any like? I think it would be fantastic if you could run it and then provide this kind of feedback you already did, so that we make sure it's correct.
That's the first of the files that you showed us today, right?
That's right. Yeah, and I show if you disable the behavior object and behavior cameras at the bottom, you should be able to start it. Karim, do you think you could do the same on the sensory motor? I don't know if any people in your lab has bandwidth or.
Well, we have currently bandwidth to analyze some data, but to be able to run an actual experiment, we still have some development to do because, as I said, not all the hardware is available yet, so we have to go and buy the components and then assemble it and test it. So it will take some time to do that, but we are going to focus on specific sets of questions, specifically pertaining to the sensory motor condition.
Okay.
It would be helpful if you, again, it's probably me who's not really into the details of how you set all these threads, but if there's a specific location where you want to initiate like a thread that we can follow on, under that project management tool, that would be great. So we know exactly where to
I see.
contribute.
I think I created a forum. Some people wrote in there, and I think I follow up on everyone's thread. Let me see. I can share my screen. Oops, sorry. That should be this one. Yeah, if you go on every page on the website has a discussion button here. And so you can create threads on any page. I think.
Oh, I'm sorry. I didn't mean threads. I meant like part of this page where you say what's in progress, what's in. This is the project management page. Is that
Yeah. This one?
Yeah. Yes. So is this always initiated by you? Like each tab is initiated by you guys? Do we have
mean,
the
if you
ability to
wanna-
initiate something in progress here?
You should be able to create tickets. If you want to, I can give you a permission to do so. Also, like it should be as easy as creating a ticket like here.
Yeah, that's what I meant, but I don't know if I have the ability to do so.
Oh, okay. If you don't, if you're, this is a public board. If you can't add tickets, you just send me your GitHub or wherever
Yeah.
GitHubs, and I'll add it as a right person.
Okay, sounds good.
Yeah. And then Lucas, you had some. I think you had some comments you wanted to make on Stimul's design.
Yeah, right. That was about the standard of power condition. I was wondering because the way it's currently designed, one has only always the same standard stimulus orientation, right? So
That's
if
right.
we compare standard, deviant, and the random control, we cannot do that for these three conditions for the same orientation. So I think Jordan Ham in his 2021 PNAS had a nice setup where he has the essentially switches the orientations of standard and deviant. In this way,
That's
you can
right.
then compare the same the three standard deviant control for the same orientation. And I was wondering if one could do this also in this protocol.
So I think we discussed this a little bit. The challenge here is statistical power, because you essentially have to double, like you switch. Think
Right.
Alex Meyer and Andrew Bastos and Jacob Westerberg, in their work with us, we actually had half of the mice that were switched.
Yeah, I
Right.
remember.
And I think Jordan's lab, because the standard oddball is faster to learn, they could do that during sessions. I think so far, what the field has shown is that the result is the same. Is that right? And so I feel like we had a conversation with Jordan's on this, and he said, given what we have shown so far, these controls are nice, but they're not strictly necessary, given what has been shown to be the same. And so I think we decided to focus on our statistical power on more oddballs types rather than more baseline types. If you are concerned about this decision, we can discuss.
There's just one additional argument for this is that currently what we have is we have different content for the deviants, right? Because we have two different stimuli that they can be the deviant, but we do not have different context to the deviants, right? If you think of the Furutashi 2024, it might be interesting to see this, check the same deviant response under different predictive context. And this way one could essentially swap the roles and
I see.
have
Well, I
the
think
same orientation in two different prediction contexts, which might
I
be
understand.
a useful thing for comparison.
I think we're actually doing that because, for instance, in the standard oddball context, we'll have omission and alt, for instance, two oddballs types that are exactly the same as in the sensory motor context. So this we are doing, I think, actually more than anybody has done before. Like we have four sensory contexts and we are switching the same oddballs in each context.
So you say that the different predictive context is either running or
That would be
the stimulus history.
the way to predict the gradings changes. In one case, you might be using the past. In the other case, you might be using the mouse movements.
Right.
So I think that would cover that ground.
Yeah, I mean, if one wants to have it comparable to this experiment or this idea, then it would be nice if it's the same category, like prediction was either predicted in this orientation or in this orientation, then it's comparing the same thing. If it's a different prediction context in the sense of modality, for example, running versus stimulus history, then it's a bit, I mean, it's also an interesting question. It's just that through a different stimulus design, we might be able to do both, essentially.
Can you use the forum to start this discussion in writing so that we can go in depth? Because I think it's a really long and important discussion. Viola, you had.
Yeah.
It's on the same. It's basically also saying if it's contained within a single experiment, it would be very helpful for us because we want to also compare to other species where we don't necessarily have the running, non-running. And then we would have the adaptation versus oddball control surprise, which would be kind of helpful for us. It would be contained. It would be about 15 minutes. It's something we can easily run in other type of animals to see cross-species comparison.
Well, I think Alex, we were planning with Andres to do that, right?
Yeah, and
Not
I think that
so we not run into that.
Yeah, so I think Andre said that they would like to try joysticks instead of a ball to get close, but it's not the same, obviously. So I didn't know that so these are not different cohorts of mice that you would do the sensory motor task and the visual task. It's just in the same individuals you will do both. Is that correct?
Yes.
But different
days.
Yeah,
different days.
Right.
Each mouse is going through 4 different types of session. It's a massive experiment where we switch. This is outlined in the website as well as in the archive paper. You can go directly in the section and double check. And please, Viola, I think there's a thread on the forum where Andre started discussing the multi-species data, and so you might want to wait there. Right. Any more comments? Everything is good. Good and small. Yes. Okay. So next week, I think we propose that we discuss the SLAP2 analysis. I think we were going to work with Carter to update the current file with the fixed timing. And we have 2 more experiments that we want to upload to Dendy. We have run with Casper and Maade as well, Andrew, a 30 minutes long experiment as well as 2 15 minutes long experiment. Those are all pilots, but in the same mouse. But there are beautiful apical and proximal dendrites Bhutan responses to the oddball design. And on one of those frees, we've confirmed beautiful receptor field mapping of the Bhutans, which I think very few people have done at this scale. So it's a really rich data set, and so I think next week we'll plan to sort of introduce what we've done on it, and then I'm hoping we can have a conversation about analysis of it. Does that sound good?
Yeah, sounds great. I have a question. So is the sensory motor, any of the sensory motor paradigms have been executed under this experiment yet, or is it still?
No.
Okay.
No, I on the SLAP2, I don't think we are planning currently to do the sensory motor experiment. I have some I'm still unclear what the behavior of the mouse will be when you first load it. So I suspect that you need to habituate the mouse to the setup to have the coupling before you start. And so
Right.
SLAP2 experiment currently are a little more in flux, so it's harder to do longer term training. Yeah, so we are focusing on the standard oddballs that is very quick to learn and it has a lot of low-hanging fruits.
Okay.
And then also the sensory motor coupling, I think it needs a little more work to be fully mature. And so the code itself, so I'm working on it.
So after the code is ready to be implemented, there's no plan to have it part of the SLAP2 setup.
Currently, no. I mean, we can discuss with Casper and others if that's worth doing.
Yeah, I just want to understand the time scale. Is it faster for us to implement things, right here where I am, and then share the outcome of that with the group, or would it be faster to get at least the first batch of sessions or experiments done with the SLAP that we can then pick up on and implement things on our end, so that we free up some time on the SLAP too for whatever you guys need to do.
I think I guess my immediate action item on this will be to figure out how long a mouse needs to be trained on the coupling to really get it. I think Georg Keller's lab has shown that they need like at least a week of training. That's what I recollect from the papers.
Yeah, it depends on the complexity of the task. But if it's just standard, coupling with a visual feedback, and you need to incentivize the animal to start moving to begin with, and that takes a few days. But once they learn that this might be associated at least with some conditioning, you're not going to be rewarding them for doing the task, but just the idea of just being in that setup and they start locomote right away takes a few days. Yeah.
I think time-wise, I would probably advocate for doing this, like figuring out this training on a cluster, like not SLAP2 instruments, like implement the bonsai coupling with the mouse, no recording to have like a clean, stable state.
Yeah.
And then if we do SLAP, that would be much later. I think that would be my intuition, because I feel like there's a lot of value in acquiring really high quality data in the standard oddball case for dendrites. Because there's a lot of things we're trying to figure out, like about data processing and so on. So that I don't want to do too many experiments.
Okay.
But so that would be later in the year if we do it.
Okay. Sounds good.
So real quick, the SLAP2, I assume half an hour. That's partly also because of photo bleaching. There's just a limit to.
We had this concern initially, and then we increased the session length to 51 minutes. In the latest experiment, which I think is the full durations. So the latest session had the total repeat number that we were planning in the perspective. So and what we still have to confirm that bleaching is not a problem, but there was activity throughout, and the receptor field block is at the very end. So the fact that we have receptor field tuning of boutons at the very end support that it worked.
Do you detrend the data?
I think Casper is doing that, yeah. Okay. So we'll meet next week with the analysis meeting on SLAP 2 and then and continue our way. Thanks, everyone. And please interact with the forum. I'll try to do my best to follow it.
Thank you. Have a good day.
Bye. Thank you.